Hello students, I am Akshay Deshpande and today we will be discussing the last class on the cell chapter, that is the unit of life, and at the end of the class we will be mainly covering from mitochondria till to the nucleus, and finally we will look into a few questions from whatever topic that we have discussed till now. Okay, and by the completion of the whole cell unit, we will try to look into some of the important questions that have appeared in the past NEET examination. Okay, in the sense, complete the 3 chapters that comes into the cell unit. Okay, so don't think that we will be discussing only the cell chapter questions at the end of the class. Okay, so after completion of the whole cell unit, we will look into all the questions that have previously appeared from 2016 to till the last NEET examination that has been held. Right, I guess we can start with the discussion today. So the first we can look into over here is the mitochondria. The mitochondria, it's a quite popular cell organelle because if you ask any student regarding about to tell about any cell organelle, every student comes up with the first name, that is the mitochondria. Okay, now why mitochondria is quite popular among the students? If you ask, generally the simple answer is it is mainly involved in the ATP production. Okay, it is mainly involved in the ATP production. Okay, hence we also call this as a powerhouse of the cell. It is the powerhouse of the cell. Now mitochondria, generally the number of the mitochondria varies from cell to cell, and even the shape also varies from cell to cell. There are multiple reasons why the mitochondrial number varies from cell to cell. The primary reason we can state that it is mainly the physiological state. Okay, it is the physiological condition or the physiological state, we can say. Why? Because not every cell is metabolically active. The ATP requirement generally varies from cell to cell. Okay, that is why we can say that the number of the mitochondria varies, and even the shape, general shape, we can say it is sausage shaped, or we can say an elongated or somewhat cylindrical. Okay, elongated or cylindrical in a shape. Okay, and generally in a normal conditions the mitochondria is not visible, and we have to use a proper stain, right? So they have mentioned that it is not visible properly until you use a proper stain. So for staining the mitochondria, we are having a stain called as the Janus green B. Okay, Janus green B it is one of the popular what we can say mitochondrial stain that is used to visualize the mitochondria in the cell. Okay, so now if we try to draw a diagram of what we can say mitochondria, this is a rough schematic representation, and this is how the mitochondria looks like. So this is an elongated elliptical shape what I'm drawing, or in some conditions, as I said, it is a quite cylindrical. Okay, not completely cylindrical, elliptical we can say, and this is how we draw the mitochondria, right? Yes, so the diameter of the mitochondria is around 0.2 to 1 micrometers, and the length will be generally varying, but we can say that it is from 1 to 4.1 micrometers μm. I have not mentioned the units, but it is 1 to 4.1 micrometers in the length. Okay, and the mitochondria is quite a special cell organelle. The reason is that it is mainly a double membraned organelle. Okay, now unlike in the case of endoplasmic reticulum, or you can consider Golgi apparatus, or even lysosomes or vacuoles, even peroxisomes or something like that, they're all single membrane structures. But however, mitochondria is a two membrane or a double membrane structure. Okay, yes, so double membrane structure in the sense you are having one outer membrane, and the next one we are having is the inner membrane, right? Outer membrane and the inner membrane, and the space next to the inner membrane, this gap between the outer and inner membrane is called as the intermitochondrial space. Mitochondrial space. Intermitochondrial space, right? And at the center, that is the inner membrane envelopes the cavity or the lumen of the mitochondria, which is mainly a dense proteinaceous mass, semi-fluid in nature. It is called as the matrix. It is called as the matrix. And also, if you observe, the inner membrane is thrown into numerous foldings that you are observing over here, and these foldings are called as the cristae. It is called as the cristae, right? Now, cristae mainly increases the surface area. Cristae mainly increases the surface area, and the inner membrane or the cristae is the site for your aerobic respiration, or we can say cellular metabolic activities. Okay, not inner membrane completely. Even matrix is also involved in the aerobic respiration process. For example, the Krebs cycle. If you look into the Krebs cycle, the site of Krebs cycle is occurring in the matrix, but however, the next part of the Krebs cycle that is entering in the aerobic respiration we are having is the electron transport chain. Electron transport chain or electron transport system. The electron transport chain, the enzymes that are required for the electron transport system are mainly present in the inner membrane or the cristae itself. Okay, so the two major metabolic processes that are involved in the generation of ATP or production of ATP are mainly occurring in the matrix, and one more it is in the cristae, right? So the enzymes that are required for the Krebs cycle, they're completely present in the matrix, but whereas the enzymes that are required for the electron transport chain, the complexes and all, they are basically present in the cristae or the inner membrane, right? Hence we have mentioned that it is mainly a double membraned structure. So we have given a fairly brief account on the structure of the mitochondria, right? So when you look into the function of mitochondria, it is the site of the aerobic respiration. Okay, so we will be studying about the aerobic respiration more in detail in the plant physiology chapter about the plant respiration, right? Yeah, so where we actually begin our discussion from the glycolysis, and from glycolysis we enter into the Krebs cycle, and from Krebs cycle we enter into the electron transport chain, right? So we will be discussing all those aspects in the plant physiology part. Now, mitochondria is quite a unique structure because you see that the mitochondria is having its own genome too, right? And it is called as the mitochondrial DNA. It is called as the mitochondrial DNA, and you also see the presence of RNA molecules, and also you find mitochondria's own ribosomes also. Okay, and generally we refer them as the mitochondrial ribosomes. And if you can see over here, this is 70S. Okay, unlike in the case of the eukaryotic ribosomes, which are 80S, but the mitochondrial ribosomes are 70S, right? So now if you look into this number, that is 70S, it does remind us of something about the prokaryotic ribosomes. Okay, prokaryotic ribosomes. So if you look into the bacteria, okay, bacterial ribosomes, even the bacterial ribosomes are also 70S. Okay, so there's a very much a similarity between the ribosomes of the mitochondria and also the bacterial ribosomes. Okay, of course we are actually having a theory called as the endosymbiotic theory, which tells us that the chloroplast and mitochondria were once upon a time free living individuals. Okay, and they had the capacity of fixing the oxygen for generation of the ATP and all, right? So endosymbiotic theory is completely an advanced level biology concept, so let us not look into it, but we can say that once upon a time the mitochondria and chloroplasts were free living organisms, but they were engulfed by the eukaryotic unicellular organisms, and they finally became the part of the cell itself. Right, so this is mainly about the endosymbiotic theory. So we can say that the ribosomes of the bacteria and ribosomes of the what we can say mitochondria are very much similar because both of them are 70S. Now, when we look into the division process of the mitochondria, they mainly divide by the binary fission, or they've just mentioned as a fission. Okay. Now, fission is actually the asexual mode of reproduction. It is mainly an asexual mode of reproduction. So this is mainly about the mitochondria. Remember, the main function of the mitochondria is it is mainly involved in the aerobic respiration, that is the Krebs cycle, mainly the Krebs cycle, and the electron transport chain or electron transport system mainly occurs in the mitochondria itself. But the difference is the enzymes that are required for the Krebs cycle, they are mainly present in the matrix of the mitochondria, while the enzymes or the prerequisite requirements for the electron transport chain are mainly present in the inner membrane of the mitochondria. I hope we have made this quite clear. Let us move on to the next one, that is the plastids. Okay. Now, plastids are exclusively found in the case of plants and euglenoids. Okay. Now, what do you mean by euglenoids? Euglenoids, we are having an example, that is the euglena, right? We are having an example, that is the euglena, right? Now, euglena is quite a special unicellular eukaryote. The reason is it is in the presence of sunlight, it can undergo with the help of produce its own food with the help of the photosynthesis, right? Yes, it can produce its own food by the photosynthesis, but however, in the absence of sunlight, it is a heterotroph. It is a heterotroph, right? So when it comes to the mode of nutrition in the case of euglena, they are autotrophic and also they are heterotrophic in nature. Okay, so they become autotrophic only during the presence of sunlight, because they have the plastids, and using the plastids they can take up the photosynthetic mechanism. But in the absence or in the partial presence of in the absence of strong wavelength of light, we can say that euglena is actually a heterotroph, and it basically hunts on small phytoplanktonic organisms and gets the nutrition by digesting them, right? But in the case of plants, there is no other alternative, hence they are completely autotrophic in nature. Plants are completely autotrophic in nature, and they exclusively produce their food by themselves with the help of the plastids, and this whole process we call them as the photosynthesis. Right? Now let us look into the classification of the plastids. Now the plastids are mainly classified into the chloroplasts, and the next one we are having is the chromoplasts, and the last one we are having is the leucoplasts. Now this classification is mainly based upon the criteria or the presence of contents. Okay, what do you mean by presence of contents? Basically what these plastid contain inside them. Okay, what kind of materials? We are mainly speaking of what materials, what kind of materials you find in the plastids, right? So based upon that we classify the plastids into chloroplast, chromoplast and leucoplast, right? So chloroplast we are all very much aware, okay, because they are the structures in the plant cell which are mainly involved in the photosynthesis, right? They are mainly involved in the photosynthesis, and the chloroplasts mainly contain the pigments, okay, the photosynthetic pigments, that is the chlorophyll A, chlorophyll B, okay, and also the xanthophylls, etc. All of these contents are mainly present in the chloroplasts. But whereas the chromoplasts are other different structures which mainly contain the fat soluble carotenoids. Okay, now examples for the carotenoids, we are mainly having the xanthophylls, right? And we are having one more, that is the carotene. Okay, carotene, right? So we all know, like many people suggest for the children, young children, to eat carrots, okay, because the carrot mainly contain carotene and beta, also called as the beta carotene, we can say. And this carotene is actually used for the production of vitamin A, and vitamin A, as we all know, it is required for the eye, right? For the good vision, in the case of eye, we require vitamin A, right? And vitamin A is ultimately produced from the carotene. Okay, this is an idea what we all know about the carotenoids. So because of the presence of the xanthophylls and the carotene molecules, substances, it gives a characteristic color, right? And the color what you see in the case of leaves, it is orange or it may be yellow, and in some cases if it is more highly deposited, you even see the presence of a red color also, right? So chromoplasts mainly contain, we can say, fat soluble carotenoids, right? Yes. The last one we are having is the leucoplasts. The leucoplasts are mainly colorless in nature, and the size are varied. You don't find any uniform size of leucoplasts, and the main function of these structures are they are mainly involved in storing the nutrients. Okay, if you ask what kind of nutrients they are involved in storing, the first one we know is the carbohydrates. So what kind of carbohydrate this structure is mainly involved in storing? It is a starch. Okay, remember it is the starch. Example for the amyloplast storing the starch is mainly the potato. Okay, potato is mainly involved in what we can say a high amount of amyloplasts which store the starch, and starch is an example for the carbohydrates. So one type of what we can say leucoplast is the amyloplast. Similarly, we are having one more type of leucoplast that is called as the ileoplasts, and these are mainly involved in storage of fat and oil. They are mainly involved in the storage of fats and oils. On the contrary, we are having elyroplasts which are mainly involved in storage of proteins. They are completely involved in storage of proteins. So now there is one important point that you people have to remember. See, in the case of plastids, we mentioned about the chloroplasts, right? And we even mentioned about the next one we are having is the chromoplasts. Okay, chromoplast. And finally, we even mentioned about one more, that is the leucoplasts. Okay, we mentioned about the leucoplasts. Remember one thing, during the developmental stage of the plants, okay, you find all these three types of plastids, that is chloroplast, leucoplast and chromoplast. But however, depending upon the requirement, the chromoplast can be converted into leucoplasts, the leucoplasts can be converted into chloroplasts, and again chloroplast can be converted into the chromoplast, and similarly leucoplasts can be again converted into what? The chromoplasts. So each of these plastids are actually interconvertible. It is what? Interconvertible. But generally you see that the leucoplasts are the ones that are mainly used for converting them into chloroplast or the chromoplasts. Okay, but there is a very strong possibility, that is depending upon the requirement, the leucoplast may be converted into chloroplast, or chloroplast may be into leucoplast, or we can say chromoplast into chloroplast, vice versa. Right? So this is one interesting fact about the plastids, that is interconvertibility. Interconvertibility. Right? Exactly. Remember this point. So now we can take a detailed look on one type of plastid, that is we are having is the chloroplast. Okay, now chloroplast is very similar to that of the mitochondria. Okay, and this similarity is mainly partly due to what we can say some of the important appearances and the functions, and also the working mechanism. Okay, mainly the working mechanism. Based on the working mechanism, we say that the chloroplast and mitochondria are quite similar, and even the origin of the chloroplast is also very much similar to that of the mitochondria, right? Yeah, why? We will be discussing that quite shortly. Now, chloroplast, if we have to draw a diagram for chloroplast, we will just draw a simple elliptical shape, okay? And it is again a double membrane structure, so obviously you have to draw two membranes for the reference. And here you find a flattened disc-like structure which are overlapped on each other in the form of a stack of coins, right? And these overlapping structures do have sometimes interconnecting structures also. And yeah, so this is how we can draw the chloroplast in the simplest way. And as I said, it is made up of two membrane. It means that we have to mention the labeling, that is the outer membrane, and the next one we are having is the inner membrane, right? And these stack of the flattened discs, what we have mentioned, they are basically thylakoids. Yeah. Now, thylakoids are very important. You will study about the thylakoids in the plant physiology, that is the photosynthesis chapter. And the thylakoids are the site for photosynthesis in a nutshell. Okay, they are the site for the photosynthesis, and the stack of these thylakoids, we call them as the grana, right? And the fluid-filled substance that the inner membrane of the chloroplast that completely covers is called as the stroma. Okay, stroma. Now, stroma is a site for production of your carbohydrates, the starch synthesis. Okay, the ultimate product of your carbon fixation, that is the C3 cycle and C4 cycle. If we ask, the starch is mainly produced in the stroma of the leaves or the chloroplast, we can say. And yeah, did we miss? Yeah, we missed one more structure over here, and this structure is the stromal lamellae. Exactly. The stromal lamellae are the structures which interconnect the thylakoids. That is what they actually do. So now let us look into the important points of the chloroplast. It's a double membrane structure. That is why we have drawn it in that particular way. It is a double membrane organelle, okay, and it is involved in the photosynthesis. Unlike in the case of mitochondria is involved in synthesis of the ATP, right? And though it is also a double membrane structure, chloroplast and the mitochondria, both of them are double membrane structure, but you can see in the diagram that the inner membrane of the chloroplast is not thrown into foldings, right? You see such kind of foldings only in the case of mitochondria, but not in the case of chloroplast. Remember this, okay? So in the case of higher plants, the main site where the chloroplasts are present are the mesophyll cells. Okay, they are mainly present in the mesophyll cells. And the mesophyll cells are the main cells in the leaf where you find highest number of the, what we can say, the chloroplasts. Again, the shape is varied, similar to that of the mitochondria. In some cases, you find lens shaped, that is mainly convex kind of shaped. In some cases, it may be completely oval, or what we can say spherical in some cases, or it may be a discoidal in nature, or even a thin thread ribbon like also. Right? So even the shape and appearance also varies. Right? Even the shape varies. The typical length, if you speak of, it is around 5 to 10 micrometers. Okay, and the width is around 2 to 4 micrometers. Okay, try to remember these numbers because they are important for your CET. You can expect 1 or 2 questions directly statement based, which of the following is true or not true. So they can mention a few points in the options regarding the chloroplasts, and you have to pick the right one or the incorrect one. Now, the number varies, as we have mentioned. Not only the shape varies, but even the number also varies. In Schleimhadomonas, it's a kind of algae. Okay, in Schleimhadomonas, you see it is a one in nature, and in some cases you can see that it is around a 20 to 14 number in the case of plant mesophyll cells. In the plant mesophyll cells, the number will be around 20 to 14 nature. Okay, now we have mentioned about the thylakoids. That is what we are writing over here, that it contains the flattened disc-like structure, that is the thylakoids, and the thylakoid which are stacked one upon the each other to give a coin stacking on each other. Okay, you remember the coins, 1 rupee coin or 2 rupee coin, if you just go and stack on each other, put one top of the another, it gives a similar appearance when observed under the electron microscope, right? And this stacked thylakoids one upon the other, one top of the other, we call them as what? The grana. Right? Now, there are also some structures, as we have mentioned, that is the stromal lamellae, which connect the thylakoids. And one similarity that we can look over here is that the chloroplast has its own genome. It has its own DNA, right? Similar to that of your mitochondria, we see that the chloroplast also has its own genome. and even it has its own ribosome, and we can see that the ribosome of the mitochondria and also the ribosome of the chloroplast, both of them are 70s. Both of them are what? They are 70s in nature. So this is some similarity between the mitochondria, okay, and the chloroplast. Try to remember the similarities and also the modus operandi, that is the functioning of the mitochondria and chloroplast. The principle involved in the chloroplast working and also the mitochondrial working is very much similar. Okay, that is called as the chemoosmotic hypothesis. You people will be studying that in more detail in the plant physiology chapter, that is respiration and the photosynthesis. Right, so I think we have made all the points quite clear over here. We have discussed all the points. So let us look on to the next part, that is the ribosomes. Okay, so here we are mainly discussing the eukaryotic ribosomes. We are mainly discussing the eukaryotic ribosomes only. Okay, don't get confused with the ribosomes of the prokaryote or the mitochondria or your the chloroplast. We are mainly discussing about the eukaryotic ribosomes. Now, ribosomes are the structures which were mainly discovered by a scientist called the George Palade. Okay, he was the person who identified it in 1953. Subsequently, he even got the Nobel Prize in Medicine or Physiology. And they are mainly made up of the rRNA. Okay, typically the ribosomes, we name them as what? The RNA present in the ribosomes, they are called as the rRNA. The small r mainly stands for the ribosomal RNA. Okay, ribosomal RNA. And you also find some amount of proteins. You also find some amount of proteins. But remember one thing, the ribosomes are special because though they are not made up of proteins completely, majority of the content is mainly the ribonucleic acid, that is the RNA, and protein content is quite less. Ribosomes show catalytic property. Catalytic property, yes. Catalytic property. What do you mean by catalytic property? Enzyme kind of property. Enzyme. They behave like enzymes. Okay. See, this is a very interesting aspect about the ribosomes because unlike if you consider any metabolic reaction occurring inside the cells, okay, you consider any metabolic reaction. Generally, you people will be studying about the glycolysis. You will be studying about the Krebs cycle, even your electron transport chain. All the enzymes, if you look closely, all of them are proteins. But here, in the case of ribosomes, they are not proteins. They are completely made up of ribonucleic acid, that is the RNA, yet they show the catalytic property. Okay, they have the capacity of bond formation. They have the property of bond formation. So if you recall what kind of bond formation they do exhibit, it is mainly involved in the peptide bond formation. Peptide bond formation, right? So this is a unique property of the ribosome. Though they are not proteins, yet they have the enzymatic property or catalytic property. Okay, and such enzymes which are not made up of proteins, okay, only the nucleic acids, RNA, we call them as ribozymes. They're called as the ribozymes. Yeah, so as we all remember, if you try to recall, in the case of eukaryotic ribosomes, they are mainly made up of ATS. Okay, they are mainly made up of what? ATS. The S mainly stands for the Swedberg unit. Okay, it is the sedimentation coefficient, what we call. Okay, so sedimentation coefficient is mainly associated with the centrifugation process, which is a technique that is mainly used for settling down or separating the cell contents. Okay, so let us not look into it more deeply. And similar to the prokaryotic ribosomes, the eukaryotic ribosomes are also made up of two units, two subunits. Okay, the large subunit is mainly made up of 60s and the small subunit is made up of 40s. Don't add them together. Remember this. Okay, please don't add the both subunit thinking that it will give us ATS. It is completely not associated with that concept, right? So like every ribosome, irrespective if it is what we can say, a eukaryotic or prokaryotic, the ultimate function, the ultimate work of these ribosomes are, they are involved in the protein synthesis. They're involved in the protein synthesis. Okay, so protein synthesis means it is the translation. You will be studying this in your molecular basis of inheritance. That is second year concept. Yeah, so it is involved in the protein synthesis. That is the main function of the ribosomes. So now let us look into another structure, that is a cytoskeleton. Okay, see, in the case of plant cells, they do have a rigid shape. Okay, they do have a rigid shape, and of course the rigid shape and structure is contributed by various factors, but one of the major driving factor that gives a plant cells proper shape is the cell wall. Okay, is mainly what? Cell wall. But in the case of animal cells, you don't find any such kind of cell wall in the case of animal cells. Remember this. Okay, so yet the animal cells, okay, yet the animal cells do show motility. Okay, they do show motility. They do have certain shape also. Okay, they do have shape, they do have motility, and this motility and shape is ultimately provided by the structure that is the cytoskeleton. It is mainly given by the cytoskeleton. Okay, now cytoskeleton is mainly proteinaceous. It means that they are completely made up of proteins. Okay, and the cytoskeleton mainly includes three types. The first one we are having is the microtubules. Okay, microtubules, you people might have heard it as the spindle fibers. Okay, if you remember from your cell division, the structures that ultimately split or separate the sister chromatids from the chromosome is mainly because of the pulling effect of the spindle fibers, and spindle fibers are none other than made up of the microtubules. Remember this. And we are having the next one, that is the microfilaments. Microfilaments, they are none other than the actin filaments. We are mainly speaking of actin. Try to recall where you have heard of the actin. If I'm not wrong, actin, you have heard about the actin in the locomotion chapter, human physiology, locomotion, right? In the locomotion, that is your sliding filament theory, right? And finally, we are having intermediate filaments. Okay, though the function of the intermediate filaments is not correctly described, okay, since their discovery, but yet we say that the main function of the intermediate filaments is they provide the shape. Okay, they mainly provide the shape and the mechanical support. Yes, they mainly provide the mechanical support. That is what they do. Right, so we have mentioned the functions over here, that they provide the mechanical support, they provide the motility, and also they provide the shape to the cell. Okay, so this is what the cytoskeleton are mainly involved in. So we have made the points regarding the cytoskeleton clear. Let us look on to the next concept, that is the cilia and flagella. Right, so flagella, we have studied about flagella in the prokaryote part, and as we all know that flagella in the case of bacteria, they're mainly involved in locomotion. Okay, but though in the case of unicellular eukaryotes, okay, unicellular eukaryotes, flagella and cilia are involved in locomotion too. Locomotion, right? Yes, only in the case of unicellular eukaryotes. Only in the case of unicellular eukaryotes. Okay, but this does not stand true for your multicellular eukaryotes. Okay, yeah. So what is the function of cilia and flagella in the case of, let me erase this. Yeah, so in the case of our multicellular eukaryotes, right? The function of the cilia and your flagella, right? They are mainly involved in movement of particles. Yes, movement of particles. Remember this. Okay. For example, in the fallopian tube, fallopian tube, right? The egg is non-motile, okay? Sperm is motile, but egg is non-motile in nature, right? So how do you think the egg actually is transported in the fallopian tube? The egg is transported from the ovary into the fallopian tube by the action of cilia itself. By the action of what? Cilia. Okay, so you can see that the fallopian tube, the cells of the fallopian tube are lined with what we can say the cilia, right? Similarly, even our ear, okay, the inner ear, that is the vestibular system, and also our cochlea is mainly lined with the cells called as the hair-like cells, because the hair-like cells are mainly having extensions on their surface, and these extensions are none other than the cilia itself, right? So cilia, in the case of the multicellular organisms, they have wide variety of functions, okay, but generally we can say that they're mainly involved in movement of particles and also trapping the particles, for example, our bronchi, okay, trachea and bronchi of the lungs, they are mainly involved in trapping of unwanted substances moving into the lungs. And that is because of the action of beating or flickering or constant motion action of the cilia, right? Yes. So it is hair-like. Sorry, there is a spelling error. It is not hair-like. It is a hair-like outgrowth. And cilia, predominantly, it is involved in the movement of fluids or cell. Yav tarad cello, for example, we are having egg is an example, right? We have just discussed over here. Fluid in the sense mucus, right? Mucus is also semi-fluid in nature, okay? And that movement of fluid is because of the constant beating action or flickering action of the cilia. So flagella are generally longer than the cilia, and they are also involved in the cell movement, okay? In the case of, we can say paramecium, okay? Or you can even say in the case of euglena, okay? They have cilia and also they have flagella. So they are mainly involved in locomotion of those unicellular eukaryotic cells. So generally, you see that the cilia or flagella, they are covered by the plasma membrane, okay, and the core is called as the axoneme, okay. So they have given the diagram in this particular way if you look into the textbook, right? So here you find two central microtubules, and there are 9 outer doublets. So 1 2 3 4 5 6 7 8 9, right? So if you can remember, this is how they have given, right? This is just a schematic representation. Please don't take it accurately, right? I'm using the pen tab and I'm not that proficient here. That is why. Yeah. So this is how it looks like. And this core is called as the axoneme. It is called as the axoneme. So here the peripheral, this part what you're looking over here is called as the peripheral microtubules, and they are 9 in number, and they are doublets, and they are doublet in nature, right? And at the center you find 2 independent microtubules, and they are complete in nature. Only there are 2, and this covering is called as the central sheath. It is called as the central sheath. You also see that from each doublet, peripheral doublets, there is a rod-like structures protruding, which is called as the radial spokes. It is called as the radial spoke, right? And also the 2 adjacent doublets are connected with each other, okay? And this is called as interconnecting bridge. Interconnecting bridge, right? Yes. So these structures ultimately help in movement, okay? And remember, for the movement, again, it is at the expense of energy. Again, ATP is required for the movement of cilia or flagella, okay? If you recall, this interconnecting bridge is actually an ATPase. It is actually an ATPase, okay? It means that it hydrolyzes the ATP to ADP and inorganic phosphate, okay? So yes, so there are 9 peripherally arranged microtubules, and the two are at the center, and this arrangement is called as the 9+2 kind of arrangement. Okay, you find that the two central microtubules are covered by the central sheath, and you find the radial spokes emerging from these 9 peripheral microtubules. Hence we can say that there are 9 radial spokes. So there are interconnecting bridge or inter-doublet bridge that connects the peripheral microtubules. So this is mainly about our cilia and flagella. So let's look on to the next one, that is the centrosome and centriole, which is very much similar to our cilia and flagella also. Now, the centrosome mainly includes 2 cylindrical structures with the pericentriole materials. Okay, it's also called as the PCM, right? It is again made up of proteins. Okay, and this PCM is mainly involved in nucleation. It is mainly involved in the nucleation. Nucleation, what do you mean by nucleation? That is a polymerization of tubulin. Okay, what happens? What do you mean by polymerization? One tubulin molecule is added to one more molecule, and similarly the tubulin molecules are added serially, and this serial structure of adding the tubulin molecules is called as a polymerization or nucleation. Okay, and this results in the formation of microtubule. It results in the formation of the microtubule, right? So the centrioles are a perpendicular and a cartwheel like organization. So if you remember your cell division cycle, cell division chapter, this is how we draw, right? They are always perpendicular. Okay, they're always perpendicular to each other, and they are mainly made up of 9 evenly spaced microfibrils, and these microfibrils are triplet in nature. Yes, unlike the doublets, in the case of your axoneme core, you find a doublet, right? Here you can see it is doublet, but however it is mainly made up of triplet microtubules, fibrils. This is how it looks like. Okay, the ultimate circular arrangement, in each arrangement there will be 9 microfibrils, but however each microfibril is made up of 3 in this way. Okay, microtubules, and the adjacent fibrils are connected also, right? The proximal part is actually called as the hub, and they are connected to the next fibril with the help of spokes. And finally, one point we have to remember is the centrioles are ultimately derived from the basal body. They are derived from the basal body. Now, let us look into the next one, that is the nucleus. The nucleus, we all know, it was discovered by Robert Brown, and due to its basic dye taking property, it is mainly stained by the basic dyes. Okay, for example, asto-orsin, asto-carmine, these are all basic dyes. They are mainly used for staining the nucleus. As they take up the basic dye, it is called as chromatin, and this naming was done by the Fleming. And the interphase nucleus is the most detailed nucleus we can say for studying. And interphase nucleus, in the sense the nucleus that is not dividing, it is called as the interphase nucleus. So in the case of interphase nucleus, you see there are 3 important parts, that is the chromatin, the nuclear matrix, and one more we are having is the nucleoli. So the chromatin is basically the DNA thread like molecules. It is the DNA. And the nuclear matrix, we are mainly speaking of a nucleoplasm, right? And finally, nucleoli are the structures, dense dotted structures present in the nucleus. It is mainly involved in the production of the rRNA, or in other terms, it is mainly involved in the production of the ribosomes. Yes. Okay, so nucleoli is mainly involved in the synthesis of the ribosome. It is the protein ribosome synthetic factory. So the next one we are having is the nuclear envelope. It is made up of 2 membranes. Okay, so apart from mitochondria and chloroplast, another structure in the cell that is made up of 2 membranes. It is mainly the nuclear envelope, and the space between the 2 membranes is called as the perinuclear space, and generally it is of 10 to 50 nanometers. Okay, so if you have to draw the nuclear envelope, this is how you draw, right? Yes. Exactly. So this is an outer membrane and this is an inner membrane, and this distance between the two membrane is called as the perinuclear space, which is around 10 to 50 nanometers. 10 to 50 nanometers. You also find presence of pores called as the nuclear pores, also called as the nuclear pores, right? And the nuclear pores are quite important because of they are mainly involved in exchange of materials between the cytoplasm and the nucleus, right? Mainly the RNA transport. So yeah, that is what the nuclear pores are mainly involved in. And nucleolus, as we have discussed, it is mainly involved in site of ribosome synthesis. And the chromosome, you mainly find the compacted chromatin material. Okay, the chromatin fibers mainly condense to form the structure called as the chromosomes. And the chromosomes, if we have to draw it, this is how the chromosomes look like, right? Yes. So this is called as a chromatid, right? And this is called as the centromere. And you also find one more structure present on this. It is called as the kinetochore. So kinetochore is the structure where the spindle fibers attach, and that pulling effect of the spindle fibers will result in splitting of the chromatids, right? And you also find sometimes another kind of constriction here. It is called as secondary constriction. And generally this constriction involves satellite DNA. Okay, satellite DNA. It's also called as satellite DNA or secondary constrictions. So based upon the position of the centromere, we classify the chromosomes into 4 types. The first one we are having is the metacentric, submetacentric, and submetacentric, acrocentric, and telocentric. If it is exactly at the center, the centromere is exactly the center, then it's called as the metacentric. If it is slightly away from the center, it is called as submetacentric. If it is at near to terminal, okay, near to terminal in this particular way, right? If it is in this particular way, we call it as acrocentric. If it is completely at the terminal, then we call it as telocentric, right? So this is mainly about the nucleus and its associated structures. So what we can do now is we can start to look at the questions. Let me erase this. Yeah. So let's look into the first question. Which of the following statement about the mitochondria is not true? And the shape and number varies depending upon the physiology. The mitochondria is a double membraned structure. The inner membrane is thrown into a number of foldings called as the cristae. And the ribosome of the mitochondria is same as the cytoplasmic ribosomes, right? So if you try to recall, the ribosomes of the mitochondria and cytoplasmic ribosomes they're different. It is 70 S, but in this case it is 80 S, right? So this is not a true statement, hence our right answer is D. Okay, so D is not a true statement. The next question. The structure that is responsible for giving colors to plants are mitochondria, plastids, Golgi complex, nucleus. So we all know it is the plastids, mainly the chromoplasts and the chloroplasts, right? So hence B is our right answer. The next one. Carotenoids are fat-soluble, water-soluble, ionic compounds, charged molecules. They are mainly fat-soluble compounds. The xanthophylls and carotins, they belong to carotenoids, and they are mainly fat-soluble in nature. So the next question we are having is which of the following statements are correct? Okay, xanthophylls are carotenoids. Yes, it is correct. Elioplasts store proteins. No, they mainly store the fat and oils. Amyloplasts store fats. No, it is mainly involved in storage of the starch. But whereas the last statement we are having is chloroplasts, contain only carotenoids. No, okay, they contain the chlorophyll and so many other different type of compounds, right, pigments. So hence the only statement that is correct are the first one, that is xanthophylls are carotenoids. Okay, along with the xanthophylls we are also having carotenes, right? So according to this particular question, A statements to be is more correct compared to any other three statements. The next question, the plant cell, in plant cell, majority of the chloroplasts are found in bundle sheath cells, hydathoid, guard cells, mesophyll cells. We all know the major content of the chloroplasts are the mesophyll cells. D is our right answer. The photosynthetic pigments are found in stroma, grana, thylakoid. Next is eleuroplasts. It is the thylakoid that mainly contains the photosynthetic pigments. Hence, we can say that thylakoid is the site of photosynthesis. So C is our right answer over here. The structure that connects the thylakoid is stroma, stromal lamellae, grana, all of the above. The answer is stromal lamellae. It is the one that connects the thylakoids. The next one, which of the following statement is correct? Thylakoids are arranged as a stack of coins on the stroma. Chloroplast is made up of two membrane, that is inner membrane, and is thrown into foldings. Okay, so this is not a true statement because there is no crystal formation in the case of chloroplasts. And the chloroplast contain enzymes for the oxidative phosphorylation? No. Okay, these enzymes are mainly found in the mitochondria, not in the chloroplasts. Chloroplast ribosomes are smaller than the cytoplasmic ribosomes. Okay, so you can see that the A and D statements are somewhat near to our correct. Okay, thylakoids are what we can say arranged as a stack of coins called as a stroma. Do you think they are called as a stroma? No, they are actually called as grana, right? Yes. So we can say that A statement is actually false statement, B is a false, C is a false, the D is more appropriate, right? So D statement is the true statement. The next one, eleuroplasts store the oils, proteins, starch, fat. It is easy, C easy question, it is mainly proteins. It is involved in storage of proteins. The next one, which of the following is a common feature between the chloroplast and mitochondria? Both are involved in photosynthesis. Okay, no, both of them are not involved in photosynthesis. Both are involved in ATP synthesis. We can say to some extent. Let us see that. Okay, and both contain an inner membrane and similar in arrangement. No. Both work on a chemoosmotic theory. Okay, yes, they both actually work on the chemoosmotic hypothesis. Okay, see, both are involved in ATP synthesis. Yes, the B statement is also true, but we can say that the mitochondria's sole function is ATP production, but chloroplast's function is not ATP synthesis solely. Okay, along with ATP synthesis, it is also involved in the carbon fixation, that is the C3 and C4 cycle. So we can say that B statement we cannot agree. Okay, we can say it is not true, and only the common feature we can consider is the both work on similar principle, that is the chemoosmotic theory. So D is our right answer. So next question, the large unit subunit of the eukaryotic ribosome is 60S, 80S, 30S, 40S. We all know it is 60S, large subunit, smaller will be 40S, right? And which completely makes us the 80S. So according to the question, it is large subunit, hence we can say it is 60S. The next question, the structure that is involved in providing cell mechanical support and motility is intermediate filaments, microtubules, microfilaments. The answer is all of the above. All of these three structures are mainly involved in providing the mechanical support and motility, right? And the next question, the cytoskeleton is made up of carbohydrates, proteins, ions, phospholipids. It is proteins. As we have mentioned, they are proteinaceous in nature. It means that they're completely made up of proteins. The next question. Cilia and flagella arise from hook, filament, basal body, tubulin molecules. They are constructed of the tubulin molecules, but they ultimately arise from the basal body. So C is our right answer for this question. The next question. The peripheral microtubules are 10 in number, 9 in number, 5 in number, 2 in number. They are mainly 9 in number with doublets. Okay. Remember this. Next, which of the following statement is incorrect? The core of the cilia is axoneme. It is actually correct. The central microtubules are 2 in number and each is a doublet. No, each of them are actually single in nature, right? They are not doublets. So we can say that this statement itself is wrong. So let us look into the last two options. The peripheral doublet microtubule is in contact with the central tubules through radial spokes. Exactly. Okay. And the central microtubules are covered by the central sheath. Yes, it is correct. So we can say that the incorrect statement is B, right? They are not two, they are 2 in number, but they are not doublet, right? So B is our wrong statement. The nucleus can be stained by Janus Greenby, acidic dyes, basic dyes, none of the above. The answer is basic dyes, right? That is why we named it as chromatin, okay? And it was named by Fleming. We have discussed that. So next question, which of the following about a nucleus is true? The nuclear envelope is poreless. The nuclear envelope is a single membrane. The nucleus contains the chromosomes. The perinuclear space is 10 nanometer. Okay, so D is our right answer. You can say the C option, perinucleus, the nucleus contains the chromosomes, right? See, the chromosomes are mainly formed during the metaphase, remember, and during the metaphase stage, the nuclear envelope is completely disappeared. It is not present at that particular moment of time. So hence we can say that nucleus contains mainly chromatin, not the chromosomes. Remember that. So the D is more appropriate for this particular question. Hence we can say D is our right answer. The perinuclear space, that is the space between the inner and outer membrane of the nuclear envelope, the space is 10 to 50 nanometers. So 10 they have mentioned, we can consider D as our right answer. The next question, the basic proteins that the DNA are associated with is histones, nucleoplasm, nuclear matrix. Last one is the nuclear pore. Histones. Okay, so histones you will be studying about more in detail in your second year, that is the molecular basis of inheritance. But remember, in the case of eukaryotic DNA, it is mainly associated with the basic proteins, that is the histones. Basic proteins are positively charged amino acid. It is mainly made up of positively charged amino acids, that is mainly lysine and arginine, histidine. These are all positively charged amino acids, and that is the more rich content in the case of histones. Okay, the principle is simple. Because DNA is negatively charged and histones are positively charged, both of them can mutually attract, and the binding between the DNA and the histones are strong. That is the principle behind the association of DNA with the histones. Let us look into the next question. The chromosome in which the centromere is at completely terminal is acrocentric, telocentric, metacentric, submetacentric. The answer is telocentric. At the complete end, if you find the centromere, then it is the telocentric. Acrocentric is somewhat near to the terminal, but not completely terminal. So we have discussed around 20 questions. I can say that by the end of this particular video, we have discussed around 100 questions. And I think discussing 100 questions for each, for this one chapter, I guess it is sufficient. And don't assume that the questions are fairly easy because ultimately the questions are going to appear from the textbook. Hence, our emphasis and our preparation strategy should be based on completely textbook. Right? So you people can start solving more and more questions on the cell chapter. And meanwhile, we can meet in next video with another chapter. So till then, thank you.